Hello, everyone. I'm Christy Risk, Senior Editor at Genomeweb, and I'll be your moderator for today. The title of today's webinar is Implementing Molecular Sample Tracking in Genomic Analysis Workflows, a case study, and our sponsor is Fluidigm. Our panelists today are Dr. Jianhong Hu, Assistant Professor in the Department of Molecular and Human Genetics at Baylor College of Medicine, and Luke Stewart, Senior Manager of Field Applications at Fluidigm. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you do not see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentations have concluded. Dr. Hu, please go ahead.
Good afternoon, everybody. My name is Jianhong Hu. I'm from the Human Genome Sequencing Center from Baylor College of Medicine. Today, I'm going to share some user experience from a high-throughput clinical research sequencing lab, how we implement the sample identification into our next-gen sequencing workflow. This is the outline of my talk today. First, I'll give some brief introduction to the Human Genome Sequencing Center and the Human Genome Sequencing Center clinical laboratory. And I'll give some description about the Fluidigm SNP Trace panel and how we implement this panel into our pre- and post-sequencing QC. And this is followed by some case studies, examples, and in the end, I'm going to sum up. The Human Genome Sequencing Center at Baylor College of Medicine, our mission is to translate the genomics-based approach to guide discovery and diagnosis for human diseases. Our center was established in 1996. We participated in the Human Genome Project as one of the several centers and completed 10% of the project. Currently, we have 180 staff. We process more than 30 sequencing instruments with multiple sequencing applications. Our sequencing instruments include 4 NovaSeq 6000 and a fleet of HiSeq sequencing instruments, 14 HiSeq X, 10 HiSeq 2000, and 2 HiSeq 2500, and also 2 MiSeq. The Illumina sequencing instruments are for the short reads sequencing, and meanwhile we have PacBio, RS2, and Sequel, and Oxford Nanopore for long-read sequencing. Furthermore, we have 2 3730 Sanger sequencing for variant analysis. So with this instrument, we can process more than 60,000 genomes per year. We have comprehensive informatics to support our sequencing, which enables us to do the large-scale, local, and the cloud computing. We also put a lot of efforts in the technology development. The clinical laboratory of HGSC was established in April 2016. Currently, we have 34 staff. We have active license for Cap Clear, Pennsylvania, New York State, California, Maryland, and Rhode Island. The current validated testing are for multiple DNA and RNA sequencing, sample preparation, and also variant validation with Sanger and MLPA. We have whole genome sequencing in validation now. Very soon, it will be implemented. The clinical laboratory has the capacity of about 36,000 next-gen sequencing libraries per year. This slide just shows two primary clinical research projects about to finish at the clinical lab. One is the EMERGE, which stands for the Electronic Medical Records and the Genomics. This is funded by NHGRI. It connects the genetic data with the electronic medical records. As one of the CAP-clear sequencing calls for this project, we completed more than 14,000 sequencing and generated more than 6,500 clinical reports. The other project is the RIGHT-10K project, which is in collaboration with Mayo Clinic. It's a pharmacogenomics project. We have completed sequencing of 10,000 clinical research samples. With multiple applications in next-gen sequencing, we have participated in many different projects, which include the common disease studies, cancer research, medical research, comparative and non-human sequencing, and of course, clinical sequencing. This slide just shows the cumulative samples we have processed in the past in several years. As you can see, our main focus is the whole genome sequencing. And we also have a good amount of whole exome and the regional capture sequencing. So altogether, in the past 8 years, we completed 193,000 samples. So as a center with such super high throughput samples, to check the samples and also to quality control the samples is very, very critical. So how we did that? So the first thing is the LIMS. The LIMS stands for the Laboratory Information Management System. So at the beginning of the project, the collaborators will receive the 2D metric tubes and the sample request form from our center. When the samples arrived in the 2D metric tubes, the samples are scanned, the tubes are scanned and validated, and the sample information are uploaded into LIMS. And then we will do the QC, and we also submit our data into the LIMS. Then from the same, the original tube, and the samples are allocated for downstream applications. At each step of the pipeline, the sample transfers are recorded in LIMS. So in addition to LIMS, which check the sample tubes physically, we also have the SNP array in the pipeline, which can check the sample DNA contents within the tube. So SNP array enables us 2 layers of sample identity check by comparing the array gender to the sample intake gender. It also helps us to compare the array-based SNP genotype to the next-gen sequencing-based genotype. So far, all the DNA sequencing applications, including whole genome sequencing, exome sequencing, and the regional sequencing, we have implemented this QC step. We have different types of array platforms, Illumina array with multiple chips, and also Fluidigm SNP Trace. Fluidigm SNP Trace was implemented in 2015. It is mainly used for the QC purposes. And the Illumina array, we also use it for the variant analysis and the copy number variation analysis. So on the right side, this table shows the sample counts. So far, we have performed by using different platforms, chips, and altogether we have more than 90,000 samples. Overall, the performance for the SNP trace is quite stable and reliable. the fail rate is less than 2%. Fluidigm SNP genotyping with the SNP Trace panel, it is an allele-specific PCR-based assay utilizing microfluidics technology. We just want to mention in here that we use it not for a clinical testing, but it's just a QC step in the clinical lab. So this panel consists of 96 highly polymorphic SNP loci, including 10 quality SNPs to assess sample quality, and 6 gender SNPs, 3 on the X chromosome, 3 on the Y chromosome, to verify sample gender. This panel is able to detect sample contamination, and it can also identify closely related individuals or samples, like family members, and we also use it to pairing two monomer samples. There are several advantages of this technology. The input DNA is low. With the pre-amplification protocol, the input DNA is as low as 25 nanogram per sample. It has a fast and a simple workflow. The samples are prepared on the 96-well plate and then transferred to the integrated fluidics circuit chips. One side for 96 samples, the other side for 96 assay primer pairs. And then the assays and the samples are processed in the IFC controller and then moved into the biomac system, which is a combination of real-time PCR machine and the fluorescence scanner. And then the data are ready for analysis. So it's kind of high throughput. On each chip, we can do 96 sites for 96 samples at the same time. So it's more than 9,000 genotyping on one chip. It is cost effective. Reagent-wise, it costs less than $10 per sample. So next slide, it shows our HGSC sample processing workflow as example for the whole genome sequencing and how SNP Trace is incorporated in our sample processing. So first, the samples are received, accessioned, and QC'd and put into our LIMS system. Then one aliquot of the samples are assayed for SNP Trace, and then the gender information is went back into the intake and then compared with the metadata. Only the passing samples are moved to the downstream processing for library construction, sequencing, and then data analysis. At this stage, the SNP array data is integrated again, and then for further analysis to compare the concordance. Not only the passing samples are used for the variant call and the subsequent data submission and sharing. For clinical samples, there's additional steps for the report generation. So in general, the whole exome sequencing or panel sequencing have a very, very similar workflow, but with just minor modifications. One thing I need to point out is that the SNP Trace sites are not located within the exome coding regions. So for our exome sequencing or panel sequencing, which mainly sequence the coding regions, we need to have strategy to integrate the SNP Trace sites into our capture panel. So for any new panels, we include the SNP Trace sites at the very beginning when we design the panel. For the existing capture designs we have been using for several years. We do not want to change the current design. So what we did is that we synthesized the probes for the SNP trace sites separately, and then we combine them with the exome or other capture panel design into the tube, mix them together, and then use it for capture and then follow-up sequencing. So in the next couple of slides, I'm going to show you how this SNP trace panel perform in the pipeline. This slide just shows you the screenshot of the SNP genotyping analysis software provided by Fluidigm. And it will show you the sample call, in general, overall call rate, and the call confidence, and whether the sample passed or failed the QC, and also give you the gender call. At the bottom in here, it will show you all the SNP performance for that sample. And in this panel, it will show you the list of individual SNPs. You can click in each SNP, and then that will give you the plot for individual SNP call. So once you get the data information, we compare with the sample intake data in here. The samples which the gender do not match, we raise a flag, and then we will have investigation on that. So this method is very effective for the batch screening for incoming samples. We can find sample like whole plate swab or flipping. When you check the individual SNP call, this assay can detect sample contamination. This slide just show you an example of one contaminated sample, screenshots for 3 different SNPs. So this SNP is either fail in the call, or it just has like a lower call confidence. So the sample contamination in general will cause the fail of the overall SNP call, or even cause like sample gender call as well. Fail in sample gender call. So this initial analysis was performed by using the analysis software provided by Fluidigm. In our center, we have a self-developed software, ERIS, which stands for the error rate in sequencing. It can analyze the sample concordance and contamination. So the ERIS use 3 sets of data, the SNP trace or any kind of like SNP array data, and the associated probe list, and the sequencing data. I'll give you a little bit more detail how it determines the concordance contamination in next slide. So ERIS queries the sequencing FASTQ file based on the SNP site list to generate the sequence-based SNP genotypes. There's some kind of like coverage requirement as well. And then this data is compared to the genotype, the array-based genotype, to determine the self-concordance ratio. And in addition, this sequence-based SNP genotype data is also compared to our database, which contains all the sample array data, and that will determine the best hit concordance. in the output file of our ERIS report, we can see the self-concordance ratio. We are also able to see the best 6 samples with the concordance ratio as well. And this flow chart shows you how the ERIS determines the sample contamination. So only the homozygote variant from the array data is used and then used to compare with the sequencing data. So if the sequencing-based SNP genotype is not homozygous, this indicates there is possible contamination. And the contamination rate is calculated by using the total contaminated reads and the total homozygous variant reads. The ERIS is a powerful tool which can identify sample swaps and duplications. So here's an example of sample swap. They are 2 samples. They showed high contamination rate and low concordance rate with self-array. And their first hit is the other sample. And this shows like a very clearly, this is a direct sample swap. This swap may happen somewhere in the processing pipeline. Any kind of problem will cause the further investigation on that. And here is an example of sample duplication. There was a time in our pipeline we have a set of samples. The first hit and the second hit of the samples are itself and another sample with almost also perfect concordance rate. And we know that the first hit and second hit samples are from different collaborators with different sample IDs. So usually we see this kind of cases for the family members or closely related members, but this is not the case for these samples. So we had an investigation on this. We contacted the collaborators, and then in the end, we found out that the first hit and the second hit samples, they are originated from the same source. The samples were sent to the collaborators, and then the collaborators sent to us independently. However, we found in here, they are duplicated samples. To summarize, the HGSC ensures chains of custody with sample quality control in research and clinical research studies. The HGSC has successfully implemented the Fluidigm SNP Trace panel into its high-throughput next-gen sequencing workflow. The pre-sequencing sample QC helps to identify the problematic samples and then remove them prior to the processing, and this can help to save the cost. The post-sequencing sample QC helps to remove the swapped or contaminated sample to ensure high-quality data. Our HGSC developed automated ERIS pipeline is able to validate sample identity and contamination. I'd like to thank Genomeweb for providing such an opportunity to present in here, and I would like to thank the leadership in HGSC, Dr. Richard Ibs, Eric Bowinkle, Donna Musny, and also my colleagues in HGSC in different pipelines, and especially thanks to the HGSC clinical lab people. Thank you for your attention.
Thank you, Dr. Hu. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel, and we will conduct the Q&A portion of the webinar after the presentations have concluded. And next up is Mr. Stewart. Please go ahead. Luke, can you hear me?
Yes, I'm coming. Can you see my screen, Christy?
Yes, we can.
Okay. All right. Well, thank you, Christy, and a special thanks to Jianhong for her wonderful presentation. I would like to take just a few more minutes of your time to review and then discuss some advancements in the sample identification application offered by Fluidigm. I'd like to start by highlighting first the journey each sample goes on throughout its testing phase. And note that when I say sample, I mean it could be blood, tissue, FFPE tissue, or even extracted genomic DNA. It should come as no surprise to all of us that each and every sample taken by a given researcher is passed through many different hands and downstream processes. For example, a histology lab, a lab for mutation analysis, or even gene expression analysis. I would like then to call out the inherent risk throughout this journey, as Jianhong has also mentioned in her presentation, which includes potential sample mislabeling errors, sample mix-ups or swaps, or even contamination of one sample with another. All of these potential errors could have a negative impact on their results. Regardless of the downstream application, every sample has a label or a unique identifier so it can be tracked and applied to results of numerous laboratory evaluations. Most often, though, the tubes are labeled manually with a marker or with a sticker with a two-dimensional barcode, and that could also be added to microtiter plates, whether 96, 384, or greater format. The physical sample location within these containers is recorded and tracked in spreadsheets, databases electronically, and even using elaborate LIMS installations. The sample identification application itself that we're talking about here adds a critical layer of quality control and confidence to sample tracking and its movements within and between labs. By genotyping predetermined SNP targets of the genomic DNA, which is present in the sample container itself. By assessing assembling and then assessing the collective genotypes of these SNP targets, a very unique fingerprint can be created and assigned to any given sample for downstream tracking, analysis, and comparisons. Fluidigm released the SNP Trace panel that Jianhong was talking about, the use in her lab, in 2014. And that panel has also been adopted by many customers. And Jianhong, she's been using that version since 2015. Within the panel, there are several key SNP target types that we include in this panel, and those include the following. Some quality SNPs, which are located in genomic areas which are sensitive to degradation. And the resultant data allows sample quality to be determined depending on performance of those specific SNP assays, for example, the call rate or the confidence scores. Gender SNPs are also included so that the gender of the sample molecularly can be identified and then quickly traced back to the sample information for a rapid identity check and identify possible sample mix-ups or swaps. Ethnicity SNPs are also included for population prediction. And by including these polymorphic SNPs that have an approximate minor allele frequency of 0.5 across 1 to 2 HapMap populations, data from those SNPs can be used to trace a sample back to African, East Asian, or European ethnicity. And we've included an additional set of polymorphic SNPs that also have a minor allele frequency of about 0.5 across those same 3 HapMap populations. And the presence of these additional polymorphic SNPs adds significant discriminatory power such that the probability of a duplicate genotype is about 1 in 3.93 times 10 to the 19th. And if you think about it, that is many, many more times than the current global population, the global human population of about 7.5 billion people. Within our newest sample identification panel, which we call Advanta Sample ID, That panel contains SNP targets located in highly expressed genes, which can be applied to whole exome sequencing studies and even RNA sequencing applications. So overall, when combined, data from all the SNP targets within each panel can be used to assign a molecular identity to fingerprint each sample, which can then be further used to identify mix-ups, swaps, contamination. You can also use that information to trace to downstream results, and you can even use it for tumor normal pairing. A brief comparison of the SNP Trace and Advanta Sample ID panels, and again, Advanta Sample ID is the new offering that we have now. A quick comparison shows that while they share the same quality and gender SNP targets, they differ primarily by the ethnicity, polymorphic, and physical locations of the targets that have been chosen. Both panels have about the same number of ethnicity SNPs, but the Advanta Sample ID panel has nearly double the polymorphic SNPs targets, and it contains SNPs predominantly, as I already mentioned, located in exons, which support whole exome and even RNA sequencing applications. Both panels also contain many SNPs that are also common in microarrays. Fluidigm has very powerful genomics tools, including our SNP Trace and Advanta Sample ID panels, and these panels and their associated kits are designed for use with our microfluidic systems, which maintain advantages in utility, confidence, and even savings over traditional techniques. These sample ID panels, which can also be customized to suit your needs, they enable this molecular fingerprinting of each sample. When applied, helps to ensure the integrity of the end-to-end sample-to-result process. Not just sample-to-result for an endpoint genotyping reaction, but from incoming sample QC the whole way through a whole exome or a whole genome process to reporting of results. The key types of the SNP targets that have been included in these panels, they enable unique sample fingerprinting with significant discriminatory power. And when you combine these panels with our automated microfluidic devices and systems such as the Juno or the Biomark HD, you can expect to realize a substantial reduction in reagent consumption. We're working in the nanoliter scale here. And also a significant reduction in time from sample to result, all compared to similar methods. Please visit fluidigm.com/sample-ID for more information. Thank you all for listening, and sincere thank you to Jianhong for her excellent presentation. Christy, you can have the control now.
Great, thank you, Mr. Stewart. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And we'll now start the Q&A. Jianhong, the first question is for you. What are some of the most frequent sample failure modes? And is it sample duplication, sample swaps, or is it something else?
I think most fail mode in our production is the sample contamination cause the fail. That will cause either the wrong call of the gender, because a client filter or not supposed to be, or the contamination caused the low QC overall call rate and it caused the sample fail.
Great, thank you. Luke, can you comment on the specific differences between SNP Trace and Advanta?
Yes, specific differences include the ethnicity and population targets that were chosen. Primarily, Advanta Sample ID is based on exonic locations. SNP Trace is primarily intronic, so one is primarily used for genome sequencing, the other is primarily for exome sequencing and potentially RNA-seq.
Great, thank you. Jianhong, how often is a failure due to poor sample quality? Are there any tips or guidelines you've implemented to help sample providers provide you with higher quality samples?
So in our clinical lab, and we have a requirement of sample quality. If the sample has low quality, we reject it. But for the research lab, we have to do a lot of samples, even with the samples with lower quality. And we have a QC when the samples arrive in here. We check the quality of the sample, and then we check in EGL. And if any samples with lower quality, we may use the protocol with the STA. That can help a lot for the low quality, low quantity samples.
Luke, are all the SNP trace targets within exon coding regions, and does SNP trace work with RNA samples?
So SNP trace is primarily, the targets are in intronic regions, but the Advanta sample ID panel has the targets in exonic regions. And yes, it could be used for RNA-seq applications. If you convert your RNA to cDNA and use that as an initial genotyping template, we have identified and used highly expressed genes in the Advanta sample ID panel itself for the targets where the SNPs reside, so you should see good representation across many different sample types within RNA-based studies.
And Luke, would Advanta work with 5 nanograms of DNA or RNA?
Yes, when you employ the preamplification or STA that Jianhong was talking about, we can go down to 5 nanograms per microliter.
Jianhong, do you have an estimate of the time or the money that you saved in your next generation sequencing workflow or genomics pipeline by incorporating molecular sample ID into your procedures?
So actually, I would say we did not have a good answer for this because we never calculate how much we saved. We just use this as a QC step to find if we have any problems, any mistakes happens in the pipeline. Of course, we saved a lot because any samples with the problem are removed, but we don't have a number for this.
And how did you come to choose SNP trace or other QC methods you have incorporated into the lab?
Yeah, before we implement this technology in the pipeline, we did a search. Yes, in the market, there are multiple different technologies can do the same thing. It's just based on the overall consideration of the cost, the flow, availability of the machine for testing. We chose this one.
Okay, fair enough. And what kind of experiments or samples did you use to validate the workflow?
To validate the workflow, usually we have multiple rounds of samples, control samples, and we do at least for 25 unique or even more, and then multiple rounds, inter-round, intra-rounds, and then to compare the individual SNP cost for individual samples. It's quite stringent to validate this workflow.
Luke, do you provide a specific protocol for Advanta with the Juno system?
Yes, we do.
Okay, great. And is there somewhere on your website where somebody would be able to find that?
Yeah, on the website, if you you can go to the Advanta Sample ID product, or in the search tab, type in Advanta Sample ID and look for the document that's listed as a protocol or a quick reference card. You could find it.
Okay,
if
great.
There are, you can also contact our support team if you can't find it.
And do you provide reference samples for validation?
Because the validation is unique to each laboratory, they provide their own validation samples. It's not something that we include in our kits for Advanta Sample ID.
Does the Advanta include all reagents or only the probes? And is the same kit suitable for both biomarkers, for both Biomark and Juno?
We include all of the reagents necessary to run, except, let me back up. It can run on both the Biomark and the Juno system. The protocol, there's a single protocol for both, and it just instructs you to use one or the other. And we do provide most reagents, including the probes. The probes come in a mixture that goes into the master mix.
Okay, great. Thank you very much. Those are all the questions that we have in our queue. Oh, no, I'm sorry, we have one more. Is there a way to easily find low-cost reference samples for validation?
Well, Jianhong, how did you identify your reference samples?
We basically use some HapMap samples. We have been sequenced before, and then we compare our validation genotyping and with previous sequencing to make sure they are correct.
Great, thank you, Jianhong, for jumping in there. And those are all the questions we have in the queue. We'd like to thank Jianhong Hu, Luke Stewart, and our sponsor, Fluidigm. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.